Thank you very much for joining us for this new webinar. This webinar is entitled When Holotomography Meets Immuno-Oncology. This is the first one of 2019, so to those who watched our previous webinars of 2018, thank you for coming back. And to the newcomers, thank you very much for your curiosity and interest. We sincerely hope that all of you will find this webinar helpful and inspiring. I also take this occasion to wish you all a wonderful new year. The webinar will be organized in 3 parts. After a quick introduction to the topic of today and to the central technique, holotomography, we will dissect the content of unique movies that we have recently produced here, in-house, with our technology. We will finish by answering 3 questions that are commonly asked. The subject of today is to discuss the new research possibilities that arise from the not-so-random encounter between the fields of holotomography and immuno-oncology. This alliance is poised to revolutionize our understanding of the immune system in general. This movie here, for example, has been obtained with our holotomographic microscope, the 3D Cell Explorer, and features colorectal cancer cells, the elongated cells, being killed by mammalian Tcells, the smaller, more intense cells. The 2018 Nobel Prize of Allison and Anjo awards the discovery of a new strategy that allows using antibodies to release the natural brakes that slow down such T-cell mediated killing of cancer cells that you can observe here in the movie, with spectacular results in melanoma and lymphoma, for example. And this brings huge hopes to people suffering from metastatic cancers. So immuno-oncology is now more than ever in the spotlight. Uncovering the fundamental details of the immune system functions and dysfunctions is crucial, and holotomography opens new territories for research, especially in the study of dynamic cell-cell interactions. For those that are not familiar with our product and holotomography in general, let me present it quickly. Our technology is unique because it works label-free and reports the 3D refractive index distribution of the cell, rather than an artificial staining signal. The extremely low light power needed to generate holograms allows for a total absence of phototoxicity, which leads to excellent time resolution over an actual virtually infinite amount of time. And we manage actually to acquire days of movies at a frequency of 1 image every 2 seconds. And if you wish to have a look, more movies are accessible in previous webinars, as well as on our Vimeo channel. This movie, for example, of a human macrophage eating bacteria has been taken for 24 hours at a frequency of 1 image every 6 seconds. I attract your attention on the fine vesicular or membrane details, to the phagocytosis of bacteria and the nuclear movement. This movie illustrates the fine resolution and contrast that the technology brings, as well as its multiplexing powers. In other terms, such movie contains a lot of information in one go. We believe that there is currently no equivalent, and I redirect you to our previous webinars to see the comparison with other label-free techniques. So how does it work? The microscope returns the distribution of refractive index in the 3 dimensions, which is the propension of a medium or a structure to slow down light. Such property is easily observable in a glass of water, where light, slow it down from air to water, induce a typical perturbation of the image of the straw that you can observe here on the right of the image. With our technology, we record such property at a microscopic scale, and we reconstitute an image of the observed cells. It becomes then possible to color the acquired volumes using the refractive index and its various derivatives, which requires the help of the computer using our software. Concretely, a coherent light beam is split in two beams called for one, the object beam that goes through the sample, and the second beam that is untouched and is rightfully called the reference beam. An interference pattern is then created when those two beams meet on a mirror. This interference pattern is then called an hologram. This hologram is finally recorded on a camera. Because of the rotating arm, holograms are collected at 360 degrees all around the sample, with a beam angle of 45 degrees, which leads to the collection of a full holographic volume describing the sample. This volume is then reconstructed into a real image by applying Fourier's theorem. Because this process is label-free and non-phototoxic, the researcher using our technology can obtain unique time-lapse imaging of even the most sensitive cells with excellent time resolution over very long periods of time. For example, you can see here a truly unique movie of Tcells killing cancer cells. To make a little bit of contrast, let me first show you the state of the art of the current and, in fact, very rare live imaging of the cells of the immune system that exist in the literature. Here play two wonderful movies from Hendrickson and Tcell and co-workers, published in 2013 and 2007 respectively in the Journal of Immunity for both of them. These movies features intravital two-photon imagings of T-cell and dendritic cell interactions. They represent an exceptional experimental achievement, as intravital two-photon imaging requires a very careful setup and present the advantage to see stained cells in their original tissue. You can also observe here remarkable fluorescent T-cell time-lapse imaging using spinning disk and light sheet microscopy that were made by Ritter and co-workers in 2017 and in 2015 in the laboratories of respectively Jennifer Lippincott-Schwartz and Eric Betzig. One can see that the acquisition length or the acquisition frequency, or both, represent limitations in these movies, and the necessity of a fluorescent tag is a source of molecular perturbations as well as a source of specific phototoxicity problems. Additionally, two-photon, spinning disk, and especially light sheet microscopy are expensive methods, to say the least, and they require special facilities and dedicated experts to handle both hardware and the huge amount of data that is produced. In this context, our technology is a nice addition to the set of imaging tools that are accessible for observing the cells of the immune system, one that will certainly open new research possibilities. Let's now have a look at movies that have been produced here in-house, and let's dissect the science that is contained within those. This movie has been acquired at a frequency of 1 image every 6 seconds for approximately 16 hours, and has been edited to keep the focus on a set of rare events. In particular, it features a T-cell mediated macrophage death that has never been filmed to the best of our knowledge. In general, the phenomenon that you will observe with us here have never been seen with such details, and we are kind of proud of it, of course. This movie features two cell types. First, the macrophages that are the large cells with broad membrane ruffles. The macrophages have been isolated from a C57BL6 mice and pre-stimulated with ovabumine peptide, as well as interferon gamma and lipopolysaccharide, to become actually an antigen-presenting cell. The main role of the macrophages is to recognize and digest non-self material within the body to present it to the immune system. They are usually long-living and mostly located to favorite non-self point of entries. You can also see a second type of cells, the T cells, which are the smaller, denser cells. T stands for thymus. since those cells mature in this tissue. T cells are central to the cell-mediated adaptive immune system and are able to kill non-self cells, such as cancer or infected cells, or also to help in the process of supporting the immune response or keeping the memory of previous antigens. Those T cells have been isolated from an OT1 mice and are naive at first, but get activated in front of your eyes by interacting with the macrophages that are presenting them the antigen to recognize. This activation event teaches the T cells what to kill actually. In the lower right corner of the movie, you can see two T cells interacting with a macrophage that suddenly stops moving and eventually dies. This is a very rare event that you just observe, and it is called T cell killing of antigen presenting cell, in this case, the macrophage. This mechanism is mediated by receptor ligand recognition, and for the details I redirect you to the work of Kaplan and co-workers published in 2000 in the Journal of Immunology. You can see the reference in the lower right corner of the screen. While the details of such interaction are still elusive, its rationale is clear. The human bone marrow can produce 10 to the power of 10 monocytes that are precursors of macrophages per day, and a similar rate of destruction is necessary to keep homeostasis. The phenomenon of macrophage killing that you just observed now is part of such homeostatic system. By now you have probably noticed the last exciting event of the movie, since the movie played already a couple of times, where the dead macrophage, that you can see right now, is removed and digested by its two fellow macrophages. Here macrophages do their main job, which is digesting and recycling dead material. What is really striking is the capacity of our technology to catch the fine details of the membrane extensions, while the cleaning macrophages are looking for dead material. Strikingly, they eventually recognize the dead cell, which trigger phagocytosis, the process of engulfment and digestion of dead and foreign material. If you pay close attention, it is possible to observe details of the dead macrophage digestion within the active macrophage. Such cellular interplays rely on complex machineries, and their inner dynamics were so far very difficult to assess. Our technology gives access to them with great contrast and resolution in space, and most importantly, in time. As a quick example, let's take the case of immunotherapies. How the immunotherapies impact the fundamental dynamics of the interaction between T cells and antigen-presenting cells, or between T cells and cancer cells, are so far very hard to assess because of image quality, the necessity of labeling, or also phototoxicity. Our technology would offer now an elegant solution to these problems. The following movie is much shorter. It has been acquired at a frequency of 1 image every 6 seconds for approximately 11 minutes. It is nonetheless remarkable as it features another fundamental interaction between T cells and dendritic cells that are in this movie slightly larger and less dense than T cells, with very active membrane protrusions. Dendritic cells are fully specialized antigen-presenting cells that bridge the innate and adaptive immune system and are specific to mammals. Much like macrophages, dendritic cells are present where non-self might show up, such as the skin or the inner linings of mucosa of the nose, lungs, stomachs, and gut. And those cells constantly probe for bits of foreign material that they could present to the immune system. You can observe unique dynamic details of this probing function in the movie, where large and rapid membrane protrusions are created from the dendritic cells' bodies in an apparent random fashion. The last movie that we will speak about today is featuring activated T cells targeting and killing colon cancer cells. Those T cells are exactly the T cells presented in the previous two movies that were activated to target the ovalbumin antigen. The cancer cells in this movie are the MC38 ovalbumin cell line, which makes them recognizable by our activated T cells. You can observe nice killings of cancer cells, and this happens through the typical Tcell MHC1 recognition. However, you have probably noticed that sometimes T cells recognize cancer cells but do not kill them. So what is the difference between a killing and non-killing meeting between a T cell and a cancer cell? So this is a fundamental question that such time-lapse imaging with our technology, coupled to accurate perturbations, of course, would definitely allow to address or start to address. This question is so fundamental that the first bit of understanding of this complex system was recently awarded by the Nobel Prize. Tasuku Honcho and co-worker discovered the PD-1 protein at the surface of T cells early in 1992 and demonstrated over 2 decades that PD-1 is a brake that slowed down the capacity of T cells to kill their target. By inhibiting PD-1 with a monoclonal antibody, Honjo and coworkers demonstrated that it is possible to stimulate the immune system of patients for killing cancer cells, with stunning results, especially against melanoma and more generally against metastatic cancers. There is no doubt that our technology could help going forward in such type of research. We have seen many movies, and you might now want to perform such time-lapse experiments for your own research. To perform them, we provide the necessary tools, hardware, and application notes to reach the same level of quality. We also detail a set of procedures to guide you through basic image analysis steps with the available free softwares that exist, which should allow you to do elegant image analysis. However, this is only a starting point, and keep in mind that image analysis is a full field of research on its own. As a proof of the interest generated by our unique technology, we can tell you that more than 100 laboratories over the world adopted it already. Preparing, we are sure, for research breakthrough and optimized drug discovery pipelines, for example. You can see here a sample of the publications that contain experiments made with our technology, and this in very different fields, demonstrating the versatility of holotomography as a technique of choice for live cell imaging. As I mentioned it at the beginning of the webinar, I would like to answer to 3 questions that we get very often on the topic. So first of all, can we easily identify immune system cells in these label-free images? So yes, it is possible using our software to differentiate T cells, dendritic cells, and macrophages because they have very different global refractive index. Second, how long could you keep immune system cells under the microscope while doing a time-lapse imaging? So the environment control that we provide allow to keep them in proper condition while imaging, like a regular tissue culture incubator, literally, because we generate no phototoxicity. There are specific application notes on that very specific point on our website, nanolive.ch. Finally, is it possible to treat cells while they are imaged? So yes, it is possible, but you need to be careful to not perturb the sample in space, in X, Y, and Z. It is possible also to equip the stage with injection devices, but we do not provide such devices. We just have a stage that can accommodate them. And obviously a proper setup is required. But if you have such project, I am sure that you are capable of setting it up. It is not complicated. So finally, you can find much more information, other webinars and application notes that show different type of research that can be enriched by our technology on our website, nanolive.ch. And with that, I thank you very much for your attention and wish you plenty of great results. And hopefully, I see you at the next webinar.